Hello, everyone. Our next speaker is our third keynote speaker, Dr. Sarah Teichmann from the Sanger Institute. Sarah does not really need introduction to the single-cell field. She has been one of the really notable leaders in the single-cell field overall, but particularly in the HCA world. And so I really like the subtitle in his presentation, Be Bold, Be Brilliant, Be Kind, that precisely describe who Sarah really is. Sarah, go ahead.
Thank you, Zemin. Thank you for the opportunity to speak here. It's a real pleasure. And as you said, the HCA has kind of been my full-time hobby sort of over the past 5 years or so. And it's really fantastic that the community has generated huge number of suspension cell data, which is what I'm highlighting on this slide, from many, many different organs and many samples from around the world. And so when the Covid-19 pandemic hit us in a really unprecedented way at the beginning of the year, what was good about the situation, about what's otherwise a tragic situation, is that the community kind of pulled together really, really quickly. And in the HCA community, the COVID-19 pandemic sort of catalyzed the coming together of the biological networks, including the lung cell atlas network. And so this is basically a community of people who are interested in airways. And of course, that's one of the major tissues that's affected by COVID-19. Not the only one, but one of the early tissues that the focus was on due to the respiratory distress symptoms. And so what happened was that the lung cell atlas biological network formed. You can find the coordinators for all of the networks on the web, and I'll come back to them later. And what this enabled was also a rapid communication across people focused on this tissue, and then we broadened it out to other tissues. And so the coordinators for lung are Martina Wayne, Pascal Barbry, and Sasha Misharin and Jay Rajagopal. And it was really out of the community response of how we can use, in the first instance, just the healthy reference data from the human cell atlas that we had at the beginning of the year to learn about the mechanisms of the COVID-19 viral infection. And what that led to was basically resources and portals for data sharing that are shown here. And then also the first HCA consortium papers that you can see highlighted in red at the bottom of this slide, which is, our first contributions, and I'm sure there are many more to come, focusing on patient tissues like you heard in the previous talk, to add to our understanding of this, the infection and the pathology that ensues. And so the way we started off as a human cell atlas community to understand the disease was to use that healthy reference data and to pull together everything that had been that was available from the community, both published and unpublished, to learn about the distribution of the viral entry receptors, so ACE2, TMPRSS2, and other proteases, and what's their cell type specific distribution across tissues. And so what happened was that Ward and Sumnak, together with Ni Huang and my group, collected all the data that we had, both from ourselves and other published data, and then reached out to the broader community, together with the Lung Cell Atlas Biological Network and support from everyone, to find out whether there were unpublished data sets for other tissues, such as the eye, the gallbladder, the heart, and so on, where there are unpublished data sets where the authors generously, very generously, rapidly, immediately, basically on the spot, shared the counts for the cells in those unpublished data sets for the viral entry receptors. And so what I'm going to focus on here is the barrier tissues, because those are the tissues that could be most important in the very early stages of infection, where the virus is meeting essentially the healthy reference data, if you can think about it in that way. And what I'll start with is the nose. And so with Martin Nawan's group, we published nasal biopsy data with bronchial and parenchymal lung data for the airways last year. And when we went back to that data, we found that there are goblet cells, so there are secretory cells in the nose that express the viral entry factors at a very high level. And this could, leads to the hypothesis that the virus may actually be sitting in the upper airway, so not just deep down inside the lung, but up at the top, and that explains some of the high transmissivity of this virus. Equally in the bronchi, lower down, we've got ciliated and club cells that have the necessary receptors, and then deep down the lung, it was known for a long time that there were alveolar type 2 cells that express the receptors already for SARS-CoV-1. There was a paper in 2004 that published this in the Journal of Pathology. So the airways here on the left-hand side have many cells that are susceptible to viral entry that we would predict from the healthy reference data could be infected, and the virus could sit inside. Now, Linda Lako from Manchester generously immediately in early March shared her eye data, and this showed us that superficial conjunctival cells, so in the top of the epithelium of the conjunctiva, express the viral entry receptors at a high level and to a lower extent in the cornea. And what this suggests is that viral conjunctivitis in could be coming through those superficial conjunctival cells, which also implicates the nasal lacrimal duct in transmission of the infection. And in the gastrointestinal tract, in the intestines, there are enterocytes that express the receptors at high levels. And then in terms of the maternal fetal interface, basically, which is relevant for vertical transmission from mother to fetus, there are cells at on the maternal side, perivascular cells, and on the stromal cells, and then on the fetal side, syncytial trophoblast cells, as well as villus cytotrophoblast cells that have the viral entry receptors. And of course, this vertical transmission is rare, but there have been reported cases, and so this is a potential mechanism for how the virus could jump across the maternal fetal interface. So this healthy reference atlas has given us some predictions and some hypotheses about where the virus could be entering. It provides some explanations about the high transmissivity due to these barrier tissues, the eye, the nose, and so on, that where the virus can be sitting, and also some potential mechanistic insights into the symptoms of the disease, like the anosmia, not being able to smell, and so on. And in this work, we also looked for co-expression patterns and found that the viral entry factors are co-expressed with innate immune factors shown down here in red. And so there's a module basically of that innate antiviral response that's already poised with the cells that have high expression of the viral entry factors. And so I just want to put this up. This is published work, so I'll just go very quickly and say thank you to everybody who contributed data, who are shown here on the right-hand side. And I mentioned some of them, Martina Wen and the other, Pascal Barbry and the other collaborators from the Human Cell Atlas Lung Biological Network, who were key in this collaboration and in interpreting the data. And then Wardon Ni and the other members of my group who worked really, really hard under a lot of time pressure to deal with huge amounts of data. And all this data is available at covid19cellatlas.org for the healthy donors, which is what I just showed you. And we're now also working with CZI to provide the patient donors from many studies funded by them, but also other studies on this website. So if you'd like to contribute and make your data available through the website, please feel free to write to CV19 CA ingest at sanger.ac.uk there. It's a way of distributing the data. So as we heard in the previous talk, the obvious next steps are to analyze patient samples. So the previous talk was blood and bronchoalveolar lavage. There are also other samples like nasal scrapings and organs from deceased patients that we can study or, as a community using cell atlas technologies to try to learn more about the mechanisms of the disease. So I'll talk briefly about nasal scraping data, and this is a collaboration with Kirsten Meyer in my program and Marko Nikolic in London, and again, Nhi and Christoph doing the data analysis. And what I just want to point out here is that from this nasal scraping data of COVID-19 patients, we do see that secretory goblet population that I presented earlier as a prediction of where the virus could sit. Those cells are the ones that have the high viral reads inside the single cell RNA sequencing data. So to confirm the prediction is it actually happens in the patients that the virus enters there. And then in addition, what we find is an inflammatory epithelial cell that has also viral reads. It's a new state that we don't see in healthy reference patients and where we have activation of innate immunity at a very high level. Now I'm going to go from the nose to the mouth and look at exciting data from biopsies from salivary glands, which are shown here, kind of on the inside of the lining of the mouth, and then gingiva, which is here underneath your teeth. And this is from the new Human Cell Atlas Oral and Craniofacial Network, which Kevin Bird and Blake Warner, Kevin Bird at University of North Carolina and Blake Warner at NIH have started. And they reached out to us, and again, Nhi Huang is the star kind of analyst of the data. What we then did was to look for the different cell populations in the oral cavity. This is the mouse at the top, and it's the human at the bottom. And just so that it's clear. And then what we did was to look for where the viral entry factors are sitting in the in those epithelial cells, and indeed where we can see them is in ductal and acinar epithelial cells. So in salivary gland ducts and the end of the salivary gland, the acini, and they are filiform and fungiform differentiated epithelial cells that express the ACE2 and TMPRSS2, and those are the ones that are suprabasal. So in other words, they're kind of in the lining of the salivary gland where the virus can actually access, not in the basal layer. And that's the prediction from the healthy reference data, and we validate that prediction with single molecule FISH. So this is data from Paula Powers and Blake Warner at NIH, our collaborators who did these staining experiments. And you can see at the top that the little gray and red dots are the viral entry factors. Pancytokeratin is showing those epithelial cells, and we can see those factors both in the ducts and the acini of the minor and parotid salivary glands, so different salivary glands in the mouth. And then if we look at COVID-19 patients to see if that prediction is correct and the virus is entering into the epithelial cells in the salivary glands in the mouth, then in red is the SARS-CoV-2 RNA. And you can see again in the minor salivary gland, the parotid salivary gland, and major salivary gland, both the epithelial cells in the ducts and in the acini harbor those SARS-CoV-2 RNA reads, showing that again, like in the nose, in the mouth, the prediction of which cell subtypes harbor the virus is correct. So I think this is really kind of, maybe it's expected, but it's kind of satisfying that the computational prediction from the data also shows that it's valid, and it helps us to understand the mechanisms of the disease. It helps us to understand that we need masks. Maybe it was understood in Asia for a long time, but it took us some months here to understand we need to protect our nose and our mouth, and it also has implications for diagnostics, because what it means is that saliva is a good or it could be an equally good diagnostic tissue as the nasopharyngeal swabs. And if we study the shed cells in the saliva, then what you can see here with the pink, where the pink brackets are, is that we can see ACE2 in the shed salivary cells, and we can also see SARS-CoV-2 infection in the bottom row. If you look at the red in the bottom row, the red markers, these are shed epithelial cells in the saliva, and we can see that the shed cells in the saliva are from the salivary glands. So they're not from the upper respiratory tract. We can show that the shed cells in the saliva are from the salivary glands mainly, and they're the epithelial cells, the pancytokeratin cells. That's what this PCR, this sorting in PCR shows us. And if we look at stainings of the SARS-CoV-2 RNA and staining of 16S ribosomal RNA from bacteria, then in these cells from the salivary glands, we can see co-infection or co-presence of both the virus and some bacteria, which is interesting. So also, again, these kind of cell atlas mechanistic studies of the cells kind of help us to understand also about what types of diagnostic approaches could be helpful. So I just want to mention in this section again that all this work was possible through intercontinental collaborations, across the whole world. Just like in the previous talk, where there was a nice collaboration across China and Israel and EU, continental Europe, and so on. And in all of this work, this also became possible through these networks of collaborations. And in case you're interested, maybe Aviv has already mentioned it in her presentation earlier in the meeting, but I just want to highlight again that for particular biological, for organs, systems and tissues, if you're interested in particular ones, please join these networks, because they're really powerful networks for collaboration, for exchange of ideas, and for driving forwards the cell atlases of these fields. And the one that I mentioned, so Kevin and Blake and so on, you can reach them through oral@humancellatlas.org. All of these are summarized at humancellatlas.org coordinators. Please join. Please feel free to suggest another network if there's a system that you're interested in that's not covered here. And I also want to just use the opportunity to again invite everybody to join HCA if you haven't signed up as a participant. And it allows you to receive newsletters, to receive discounts from companies, and to be part of the registry for the Human Cell Atlas. So in the last part of this talk, very quickly, I'm going to talk about a couple of other types of tissue samples, blood and organs. So I've talked about the living patient biopsies and tissue, and I will talk very quickly about COVID-19 organs and blood. So in terms of samples of cells from COVID-19 patients, we're generating a lot of single cell data from blood, as you heard in the previous talk, and I'll present later. So almost 4 million cells now, and then much less from tissues. These are data from Boston from deceased COVID-19 patients, so nuclear data. From autopsy tissue, which I'll just mention briefly, what we have available is many different tissues, of course, from these COVID-19 autopsies. And here you have an example from the lung, where we can see very variable loads in different regions of the tissue. So there are some lung regions with low virus, but other lung regions with high virus. So it's you can see these very distinct patterns in the white and in the yellow box of the lung. And from these autopsy samples, there's of course many other tissues, kidney, heart, and so on and so forth. And Omar Baraktar's group, in collaboration with colleagues in London and Newcastle, is looking at many different tissues from the point of view specifically of the vasculature. And so that's a very interesting approach to understanding end-stage disease from these deceased patients. Of course, from the living patients, we can follow kind of all different levels of disease in terms of blood, peripheral tissue, and here in the UK, we have a network that covers Cambridge and Newcastle with a network of collaborators shown here. And this network kind of got together through a real team science approach that includes the clinicians who are gathering the samples, as well as us on the genomic side, and then computational scientists from Marioni and colleagues and so on, working together, having lots of Zoom meetings to discuss the study design of the patient. So we have about 130 patients here from different levels of disease, asymptomatic, mild, severe, critical, and so on. and we've profiled them with the total-seq antibodies. And so what that gives us is the distinction between the cells at the mRNA level, also at the protein level, and we can then calculate the changes in cell proportions. And I just summarize very quickly here that we see there's 3 cell DCs and CD8 T cells going down, but then platelets, proliferating monocytes, and CD14 monocytes here going up on the bottom. And if we look at gene expression patterns, then we can associate sort of signatures with clinical severity levels. And so in the plasma cells, there's more stress factors with increasing severity in proliferating monocytes. There's more of these cytochrome oxidases, these enzymes with severity going from left to right. And we can look for gene expression signatures that are associated with worsening clinical score, like these Fc gamma receptor RNase 2 and so on in the proliferating monocytes, where you're going from worse on the left to better on the right. Now, if we drill into the lymphoid compartment, then if we take together the protein and the RNA markers, we can distinguish many different subpopulations, like you heard in the previous talk, different, for instance, CD8 T cell subtypes. And it's the effector memory cells here that I'm pointing to that we see particularly in the COVID-19 patients with clonal expansions. And we do see motifs that are shared across COVID-19 patients in those clonally expanded CD8 effector memory T cells. And these motifs in the beta chain of the TCR are shown here. And we assume that these are to do with the PMHC recognition of the SARS-CoV-2 because they're from clones that are expanded in the COVID-19 patients, and they're shared across several donors. So the number of donors with the motifs is shown on the y-axis, and the log 10 score, the negative log 10 score of the significance is shown on the x-axis. And so all of these motifs are shared across many patients and are highly significant, across many cells from many patients. So we hypothesize that these motifs are T cells that are responding to the PMHC from the SARS-CoV-2 virus. Although we haven't used tetramers to purify them. So this is potentially interesting. So just to summarize here, I've told you about studying using the Healthy Reference Cell Atlas to predict which cell types are susceptible to infection, and then studying nasal scrapings, oral biopsies from COVID-19 patients to show the cells that are actually infected, and then a little bit of work from the UK on blood cohorts and a little bit on the organs of deceased transplant donors. I want to just finish by advertising jobs in my program. So we're looking for new faculty members. If you're interested in joining us, please go to jobs.sanger.ac.uk or just write to me. So this is at all levels. It's not just junior level, also senior level faculty. And I will finish there and say thank you, and I'll take any questions.
Great, thank you very much, Sarah, for the wonderful progress, especially in the COVID-19 space. So let's see, we have a few minutes for questions. Let me start by asking the viral presence in multiple types of cells, because initially people are under the impression that the virus only infects epithelial cells because they express ACE2 and TMPRSS2. But I think based on I believe one of your talks slides show the presence of viral sequences not in a small number of cell types, actually in a very large number of cell types. You can detect viral sequences. I believe in this morning's talk by Aviv Regev, she also showed there are presence of viral sequences in more than just epithelial cells. So, I mean, what's your take on those? I mean, what are those real infections, or those are bystander infections? And presumably many of those don't really.
Just from the. So from the, if from the ACE2, from the viral entry factor patterns, if we just look at the healthy reference, they're not only in epithelial cells. I mentioned also in the decidua those fibroblasts and perivascular cells, the pericytes, if you remember. So in the endometrium, in the pregnant form of the endometrium at the maternal fetal interface. So these are perivascular cells and fibroblasts that have the factors. And in fact, we see them in other tissues, like in fibroblasts and perivascular cells in the heart too. And then I also mentioned, the trophoblasts and so on. So it's not only epithelial cells. I was emphasizing the epithelial cells, because I was talking about the nose and the mouth, and in those tissues, it's mainly in those cells. But there are also other subtypes, and there are lower levels in the heart and the cardiomyocytes, but the higher levels that we see in our data in the heart are perivascular and fibroblasts.
How about those different immune cell types? I remember we've particularly highlighted the present virus sequences in macrophage cells, which can explain by maybe the kind of interaction. Maybe they're eating some other cell types, but there are probably the presence of other immune cells, and my recollection is that they don't really express those.
No, they don't.
EC2 or temperance. So
no.
The question is.
There are two possibilities, right? One possibility is there's a different entry pathway, or the other possibility is that it's ingestion, like you said. So it's eating. Yeah.
Okay. Yeah. All right. So there is. You also mentioned the co-staining of bacterial sequence signal and the viral signal there. So that's I'm trying to figure out what that really means. So do you think somehow the presence of bacterial species there somehow would enable the viral infection? Maybe the human cell types don't necessarily have the entry point for the virus, but somehow the presence of bacteria will somehow enable those. I don't know if you're implying this or not, because you quickly went over those.
No, yes, yeah. I wasn't implying that. So the way I think about it more, because those are cells, those suprabasal cells in the salivary glands, they do have the viral entry factors. So, in that sense, mechanistically, it's not surprising that you see the virus there. It's just interesting, that the prediction is correct. That the bacteria is there. So my interpretation was more that these are damaged cells, and that therefore they have multiple susceptibility. So that was my interpretation, but we don't really know. I just want to show it because we have the stainings.
Great. Also,
what it also says from a diagnostic point of view, is that saliva is a really powerful diagnostic tissue, I think.
Great. Also,
we have more data on that I didn't present.
You did call for maybe participation in the HCA. Maybe someone can contribute to samples or effort. Lots of people, would love to join. Except that, right now, various countries, including China, but not necessarily limited to China, there are multiple countries have more stricter rules on the genetic material sharing. I don't know if you have, when you are working with people all over the world from different countries, do you encounter
this is all so this is just the expression matrix, okay? So we don't share any raw data. So it's simply the expression matrix.
Okay.
It's not identifiable per se. It's not identifiable data. So I don't know whether that makes a difference from a legal point of view for you.
Okay, got it. So let me ask a final question. This question from Guoji Guo. He's asking that ACE2 expression in lung is relatively low. Is there any other potential viral receptor?
So I think that the ACE2 expression, even if it's low, is very predictive of viral entry. So that's my opinion. Because what I've seen, for instance, in the salivary glands is expression is very low. We only detect it in a small percentage of cells, but those are the cells where you see the viral RNA. It's those cells. Same in the nose, although that level is higher in the nose. So I think that, yes, single-cell RNA sequencing data has a limited sensitivity. So Guoji's right. Sometimes these levels are very low, but from what I've seen, it's very predictive.
Okay.
Okay.
Great. Yeah. So our time is up. I want to thank you again for the wonderful presentation to the AC.
Thank you, Zemin. Thank you, everybody.
Great.
It was a pleasure.
All right.
Thank you.